This is your go-to student podcast where we take complex science and make it sense. So I am Nazira.
I'm Nurshamira.
I'm Afifah.
I'm Binti, and we are 4 biotech students who recently worked together on a portfolio for our functional genomics and proteomics.
So today we will be exploring the roles of functional genomics and also proteomics in ecosystem health, bioremediation, and environmental conservation. It is quite mouthful, but trust us, it's gonna be pretty exciting stuff.
That's right, Nazira. The 21st century has brought us rapid industrialization, which has been great for economic growth, but also comes with serious environmental consequences.
Exactly, Shamira. Issues like limited access to clean water, poor waste management, and climate change have become urgent concern to government, industries, and communities worldwide.
Industry play an important role in our economy, but unfortunately they also contribute significantly to pollution. They often release untreated or partially trapped hazardous waste that contains harmful chemicals which damage soil and water quality and threaten ecosystem and human health.
And the problem is worsened by the use of cheap and non-biodegradable chemicals in industrial processes. These chemicals linger around in the environment and add to pollution.
But here's some good news. Bioremediation is emerging as a promising solution. This method uses microorganisms, plants, or enzymes to detoxify contaminated environments, essentially nature's own cleanup crew.
With advances in technologies like metagenomics, scientists can now study microbial communities in great detail. And these microbes play an important role in breaking down persistent pollutant in soil and water.
Since environmental issues cross border and extreme, require integrate multidisciplinary approach methods like mass exploration, sequencing, protein and metabolic profiling, and bioinformatics help us understand the carbon rules.
So, omic approaches like metagenomics, metatranscriptomics, metaproteomics, and metabolomics are providing critical insights into how microbes function and interact with polluted environments.
These tools are so effective that agencies like the Environmental Protection Agency are prioritizing them to monitor and optimize bioremediation efforts to a polluted site.
By identifying key enzyme and regulatory elements, molecular methods have developed more sustainable and efficient environmental management strategies.
Rapid industrialization has led to serious environmental problems, from polluted water and poor waste disposal to climate change and toxic chemicals. To address these challenges, scientists are turning to bioremediation, a method that uses microbes, plants, or enzymes to clean up pollution naturally. Thanks to omic technologies like metagenomics and proteomics, we can now track how microbes respond to pollutant and optimize cleanup efforts. With supports from agencies like the EPA, these tools are shaping a more sustainable approach to environmental management.
So, if you are curious on how tiny microbes help environmental cleanup, you're in the right place. So let's dive deeper into this fascinating world. Okay, so picture this. Our planet is under pressure, from choking pollution to mountains of waste and a climate spiraling out of control. These aren't just headlines, they are our reality. Yes, government and NGOs are stepping up, but if we are being honest, the real game changer, it's science. So it's both innovative and unapologetic science.
Exactly, and that's where omics comes in. Genomics and proteomics give us tools to look beyond what we can see. Think of them as microscope and stethoscope for ecosystem.
Oh, I like that. Yeah, instead of just seeing that fish in the river, we can actually figure out why they are dying and what is happening at the molecular level.
Even more than that, guys, we can act before it gets too bad. And that's the magic of early detection through protein and DNA.
Yep, that's right. So let's break down to the project and walk our listeners through what we discovered.
Okay, so let's start with genomic first, especially how it helped us to study about microbial communities. So microbes are everywhere. They are in the soil, water, and even the air. And using metagenomics, we can study all the DNA from an environment without needing to grow anything in a lab.
That sounds really efficient, so we can just get the soil or water sample and study what is everywhere.
Exactly. With 16S rRNA gene sequencing, we can figure out what kinds of microbes are present, and if we use shotgun sequencing, we can also see what functions they are capable of and what genes they carry. So we can actually see what the microbes are doing. For example, some of the microbes have gene that helps to break down pollutants like oil, heavy metals, and plastic. Metagenomics help us to find these natural clean-up crews in places like contaminated soil or wastewater.
To better understand what microbes are doing in the environment, scientists use methods like 16S rRNA gene sequencing to identify them, and shotgun metagenomic sequencing to explore their genetic functions. These techniques reveal important details about microbial metabolisms, resistant mechanisms, symbiotic relationships, and even how microbes adapt over time. To handle all this complex data, scientists rely on bioinformatic tools and databases like MGRES, QIIME2, and Cake. These platforms help to process, annotate, and interpret sequencing results, linking microbial genes to real biochemical pathways.
By saying that, Shamira, this help with environmental cleanup, right? Like bioremediation.
Exactly. It helps scientists to know which microbes to use and where. Plus, genomics can even track changes in microbial communities after pollution or other environmental changes. It's like an early warning sign for ecosystem health.
That's amazing, but genomics isn't just for microbes, right?
Not at all. Another big area is biodiversity monitoring using environmental DNA or eDNA. This is DNA that animals leave behind in the environment, like from skin cells, hair, and feces. So no need for nets, trap, or even seeing the animal. With DNA barcoding and metabarcoding, we can identify species from the samples and even monitor the whole ecosystem.
Genomics plays a key role in tracking biodiversity through environmental DNA or eDNA, which are genetic material left behind by animals in soil, water, or air. This method helps detect multiple species without capturing or disturbing them, perfect for monitoring rare or endangered wildlife. Tools like DNA barcoding and metabarcoding make species identification faster and more accurate by comparing DNA to global databases like BOLT and GenBank. With next-generation sequencing, eDNA now offers a fast, ethical, and cost-effective way to study ecosystems in real time.
That sounds perfect for tracking endangered or hard to find animals.
It really is. eDNA has been used to monitor fish in the rivers, detect invasive species, and even track insect and pollinators using air sample in the forest. On top of that, genomics can show us genetic diversity within the species, which help us in understanding how populations are doing and how they might respond to things like climate change.
Genomics becomes even more powerful when combined with tools like GIS, remote sensing, and ecological modeling. This integration helps scientists map species distribution, predict habitat changes, and forecast biodiversity under climate or land use shift. Beyond research, these insights support conservation planning, habitat restoration, and sustainable natural resource management. It's how Genomics is shaping smarter environmental decisions for the future.
It's truly a powerful tool. It helps science, conservation, and even pollution cleanup all at once. So, we've been talking about pollution, stress, and all the damage our environment is soaking up. But now, let's zoom in and go microscopic. I am talking on a cellular level. This is where proteomics come in, and I know it might sound like a heavy term, but it's actually pretty fascinating. Alright, Afifah, so this is your moment. Time to hit us with science.
With pleasure, Nazira. So, proteomics is all about studying protein, the workhorses of the cell. While Genomics tells us about the blueprint, proteomics show us what actually happening inside the organism.
So it's like saying who's copying and doing the work.
Exactly, Ken. And when it's come to pollution, proteins are the first one to respond. And certain proteins act as biomarkers, which means they let us know when organism is stressed or exposed to harmful chemical.
Which proteins are we talking about?
So, Shamira, now we are talking about heat shock protein. They play a very important role in protecting our cell, especially when an organism is under stress. Then we have cytochrome P450, which is essential for breaking down toxin in our body. And let's not forget about metallothioneins. This protein bind to heavy metals and help in lessen their harmful effects.
And these changes happen even before you see the physical symptoms.
These changes in protein happen in long time before you see any visible symptom. That's why proteomics is really important. It helps us to find signs of stress pollution early, almost like noticing an illness before you can see any obvious sign.
Certain proteins in organism act as biomarkers, warning sign of pollution or stress. For example, heat shock protein increase under stress. Cytochrome P450 enzyme help break down toxin, and metallothioneins bind to heavy metals. When level of this protein arise, it often means the organism is trying to protect itself from pollution. Scientists detect these changes using proteomic tools like two-dimensional gel electrophoresis and mass spectrometry.
What about non-modern organisms? Can proteomics still work?
Okay, very great question, Ken. It does. Even if we don't have full genome data, we can still use RNA sequencing to build a protein map. One study even found over 1,000 protein in a marine organism from polluted site just from proteomics alone.
Wow, that really opened doors for environmental monitoring.
It really does, Shamira. And when organisms are under multiple stressors like pollution and heat, protein related to metabolism and reproduction can drop while stress response protein rise. Proteomics focuses on proteins, the molecules that actually carry out works in cell, not just the instruction written in genes. Using tools like mass spectrometry and two-dimensional electrophoresis, scientists can check which protein increase or decrease when organisms face environmental stress. This gives a more accurate, real-time picture of how organisms respond to pollution or other stresses at the molecular level. Proteomics is just not about single species. With metaproteomics, scientists can study entire microbial communities in environments like soil, oceans, and sediments. These microbial proteins show how communities respond to pollution and climate change. They also reveal how ecosystems carry out essential tasks like nutrient cycling and breaking down organic matter. It is a powerful way to understand environmental health on a deep level.
That paints such a clear picture of the ecosystem health. Okay, Ken, you tackle remediation. how do these omics actually help fix things?
Here's where it gets specifically. Functional genomics help us pinpoint microscopic genes to degrade pollutants like minerals, pesticide, and heavy metals. These genes in the mono-oxygenase and oxygenase are key players in breaking down these compounds. With metagenomics, we can scan contaminated environments and find microscopic for cleanup. Scientists can even use contaminated genomes to select or engineer strains that work faster and more efficiently.
So that's the gene level, but how do we know if those genes are really active?
There's proteomic claims. It checks if the cells are actually expressing those genes, for example, the classification enzyme or sequence stress protein tell us that the person corresponding actively breaking down the protein is like going from knowing what is possible to knowing what is really happening.
So my question Ken, is omic give us more real control in real time?
Yes, proteomics give real-time feedback. Instead of waiting for visible result, we can track ion expression to optimize things like pH, temperature, or temperature to improve the replication process. Even in replication, proteomics help monitor how plants respond to stress or heavy metals, showing whether they are thriving or lower.
So it's not just trial and error, it's precision guided process.
Exactly. This will let us make the best decision, monitor progress, and correct anything. It's not just about removing contaminants, but restoring the entire ecosystem more successfully. To me, that's the real power of using genomics and proteomics that turn environmental recovery into something measurable and meaningful.
Promised technologies like genomics and proteomics help track the success of bioremediation by monitoring biomarker protein and microbial activity over time. This molecular insight helps scientists fine-tune clean-up methods, ensuring long-term effectiveness with minimal impact on the environment. By combining functional genomics with proteomics, we can plan smarter, more sustainable solutions that support real ecosystem recovery.
So, wrapping it all up, what was your biggest takeaway from this project?
For me, it's how invisible things can tell the biggest stories. A teaspoon of soil might hold the key to change an entire river.
For me, is the early warning power of proteomics. We can detect damage before it's visible. And how do you guys think? How cool is that, right?
And I love learning how we can not only study ecosystem but extend using this knowledge. Real world impacts place in the world, right?
Absolutely. And for me, the biggest lesson was how powerful collaboration is between disciplines, tools, and even people like us. This project showed that we need each other's strength to make science meaningful. Well, that's a wrap up for this episode of Omics Unplugged. We hope you enjoy learning about how genomics and proteomics are shaping the future of environmental science.
If you are a student like us, don't sleep on this topic because they are going to be huge in research, conservation, and even industry.
And if you're curious, keep asking questions. That's how science grows.
Thanks for listening, and don't forget, sometimes the tiniest molecules tell the biggest story.
Okay, so till next time, take care and stay curious. Bye!